In this video, we're going to learn about nucleic acids. We'll learn about the structure of DNA and the structure of RNA and find out how those two molecules are similar and how they're different from one another. So nucleic acids are macromolecules that store our genetic information, and they are the only macromolecules that can reproduce. And this capability allows for the growth and repair and reproduction of organisms, which is very important if we want to continue to stay alive and pass our genes on to the next generation. So the basic subunit of nucleic acids are nucleotides, so they're the building blocks. And a nucleotide is made up of 3 parts. First of all, there's a pentose sugar or a 5-carbon sugar, which could be either ribose or deoxyribose. So RNA has ribose and DNA has deoxyribose. That's where the first letter of their word comes from. Then the second thing they have is a phosphate, so a PO4 group combined to the last carbon in the sugar, so the 5th carbon. And then the final thing that it has is a nitrogenous base. And you may have heard of 4 of them before when you learned about DNA in grade 11. So adenine, thymine, cytosine, and guanine. There is a 5th nitrogenous base that you only find in RNA, which is uracil or U. And below here you can see when we combine all 3 of those components, the schematic sketch would be what you see just right here. So we represent a circle for the phosphate, pentagon for the pentose sugar, and then usually a rectangle with the letter inside of it for each of the nitrogenous bases. The individual parts of the nucleotide are combined through condensation reactions or dehydration synthesis. First of all, we're going to talk about where those bonds are formed. So we need to take a step back and remind ourselves of how we number the carbons in a sugar. So if you remember, this part at the top would be the oxygen, and that's the most important functional group in the ring structure of the sugar. So we number everything from that important functional group clockwise around the ring. So this is carbon number 1, and that's where the nucleic acid is, the nitrogenous base is combined. And then this would be carbon number 2, number 3, this is carbon number 4, and then the phosphate is joined to carbon number 5. Now, if we were joining multiple nucleotides together, you would join it from the phosphate on carbon number 5 to the 3rd carbon on the sugar. So you would have your phosphate joining here, and then you would have another sugar, and then the nitrogenous base, and that would just keep continuing on and on and on down the molecule. So DNA is a double-stranded molecule, so the 2 strands actually run anti-parallel from one another, and they're held together through hydrogen bonds between the nitrogenous bases, and those bases are called base pairs. So you would have adenine forming hydrogen bonds, actually 2 hydrogen bonds with thymine, and cytosine forms 3 hydrogen bonds with guanine. And these 2 pairings are called complementary base pairs. So adenine is complementary to thymine, and cytosine is complementary to guanine. So this is a very short section of DNA, just to represent that anti-parallel arrangement and the hydrogen bonding. So you can see here that adenine and thymine are forming 2 hydrogen bonds, and guanine and cytosine are forming 3. You can see that in this case, the sugars and phosphates are pointing up, and in this one, the sugars and the phosphates are pointing down. We also say that the 2 strands run anti-parallel to one another, but that also means that one is running from 5' to 3', and the other one is running from 3' to 5'. So what this means is this, remember, is the 5th carbon, so the phosphate. So the part that's pointing up at the top is the 5th carbon, and the point that's at the very bottom down here is the 3rd carbon. So that's said to be the 5' to 3' strand. The other strand is upside down, so this is the 3rd carbon, because you count 1, 2, 3, And down here is the phosphate, so that would be the 5th carbon. So this one runs from 3' to 5'. That 5' to 3' direction and 3' to 5' direction is very important to understand, because we are going to be mentioning that a lot when we talk about DNA replication, transcription, and translation, because the enzymes involved in those processes can only work in certain directions, and that direction is the 5' to 3' direction. So RNA differs from DNA because it is a single-stranded molecule, and it can't be built by itself. It actually has to be built from DNA. So there's instructions on DNA to actually build RNA, and then that RNA can be used to build protein. So we'll talk more about how that all happens when we learn about transcription and translation. And differing from DNA, where the nucleotides are adenine, thymine, cytosine, and guanine, RNA nucleotides are adenine, uracil, cytosine, and guanine. So instead of having thymine, we will insert a uracil where a thymine should be. And the other thing that's different with RNA is its sugar is ribose instead of deoxyribose. And the only thing that differs between ribose and deoxyribose is that on carbon number 2, there is a missing oxygen on deoxyribose. So that's where the deoxy comes from. So there are a few other nucleic acids that don't fall into the category of DNA and RNA, and we've actually talked about those over the course of our unit in our metabolic processes unit. So ATP, and ATP and AMP, are all energy carrying molecules. ATP, if you remember, drives all our energy requiring reactions in the cell. NAD plus and FAD, if you remember, are from cellular respiration, and they become reduced, and then they donate their hydrogens and electrons in the electron transport chain to drive the production of ATP. And then finally, NADP plus is another coenzyme that's found in the electron transport system of photosynthesis, and it becomes reduced, and then it donates its hydrogens and electrons to convert carbon dioxide to simple sugars. So DNA supercoils to kind of wrap it up into a more condensed form. So I like to think of it as, when you wrap up your Christmas lights on, a piece of cardboard to prevent them from getting all tangled. Same idea for DNA. So the way this happens is every 200 nucleotides of DNA will wrap around 2 times around a set of 8 stabilizing proteins that are called histones. And that 200 nucleotides wrapped around those histone proteins is called a nucleosome. But the coiling doesn't stop there. Those nucleosomes then coil on themselves, and then on themselves, and on themselves again to form chromatin fibers. And these fibers condense and coil even further. And at that point, the DNA is now called supercoiled. Now, in DNA, there will be repeating sequences that aren't part of genes, so they aren't coding for proteins, but they do have important roles. So one of these is called VNTRs, or variable number tandem repeats, and these are repeating sequences of DNA, and they're found in the introns, so as I mentioned before, the non-coding regions of DNA. And these VNTRs can vary in length between individuals, which can be helpful in figuring out the difference between one person and the next, because that may be the things that make us more different. So an example would be like CAT repeating over and over. In one person it could repeat 10 times in that section, and in somebody else it could repeat 15 times. Now, if you remember from grade 11, we learned about this, the structure of a chromosome. And if you remember, the centromere is that condensed bit of DNA that holds 2 sister chromatids together after replication has happened. So you can see that in this illustration. Finally, a special part of DNA on chromosomes are called telomeres. And telomeres are long sequences of repeating nucleotides, and they're found at the ends of chromosomes. And when cell division happens, a small bit of that telomere can break off every time. And eventually, if that telomere is completely gone, the cell can no longer divide. Because once it starts dividing and the telomere is not there, parts of the coding regions of DNA would break off, and that would be really problematic.